Having seen gametogenesis, now we are going to see about fertilization. Fertilization is the fusion of male and female gametes. These two haploid gametes, they join together and they form a single cell zygote, which is diploid. So now let us see the various stages involved in fertilization. First is the arrival of the sperm. See, fertilization is happening in the female genital tract, but spermatozoa is produced in a male individual. So the sperm has to arrive the fertilization site, which plays a major role in fertilizing. So first, the semen is deposited in the female genital tract, that is in the vagina, and this process is called as insemination. At each ejaculation, around 300 million spermatozoa are released into the vagina. How the sperm moves to the ampulla of the fallopian tube? As you know that the sperm, it has a tail, and the lashing movement of the tail of the spermatozoa, it will push the spermatozoa forward. The spermatozoa which are released in the vagina, they travel through the uterus and they moves up into the fallopian tube. So the sperm has to manage to enter the fallopian tube and to reach the ampulla. For this, in the beginning, once the semen is deposited, in the beginning, the contraction of the uterus and the fallopian tube, it helps the sperm to move in. They contract, the uterus and the fallopian tube, they contract, so they help the spermatozoa to move in. But in later stages, the sperm, it has to swim on its own. And for this reason only, if you are able to recall, we have seen that for a normal fertilization, 60% of the spermatozoa should have a normal shape, out of which 40 should be having high motility. This is for this reason only, because if the spermatozoa are not able to swim, then they will not be able to reach the fallopian tube where the egg is waiting. And the speed of the spermatozoa is 1.5 to 3 millimeter per minute. In the vagina, there are leukocytes. In the female genital tract, there are leukocytes which engulf the spermatozoa enormously. So, only a few spermatozoa, a few thousand, out of these millions, only a few thousands will be able to reach the fallopian tube. Right. Now that we have seen the arrival of the spermatozoa, it has to fuse with the egg. So the egg has to reach there. How it's happening? Let's see the arrival of the secondary oocyte. See, this is the ampulla where the egg and the sperm, they are going to fuse to form the zygote. The secondary oocyte is released from the ovary. This is the ovary, so it is released from here. This will be caught like this by the fimbriae and it will be pushed into the infundibulum. Once it reaches the infundibulum, you might have studied in the earlier classes, ciliated epithelium, which is present in the respiratory tract and in the fallopian tube. The cilia, they moves and they pushes this secondary oocyte here towards the ampulla. And in the ampulla, the egg is surrounded by numerous spermatozoa. But only one spermatozoa, it succeeds in entering into the secondary oocyte. Once the spermatozoa enters, as we have seen in the last class, it induces the completion of the meiotic division. So, after the entry of the spermatozoa, the secondary oocyte, it gets converted into an ovum. So if the question is with the options, say for example the question is, the sperm enters into dash ovum or secondary oocyte. The answer is, it enters into the secondary oocyte. After the entry only it gets converted into ovum. Now there is an interesting process happening within the female genital tract, which is called as the capacitation. That is, the female genital tract, it secretes certain substances which removes the coatings on the acrosome. This is the acrosome. There will be some deposits over the acrosome. These secretions, they remove these coatings. Because of this, the receptors in the acrosome, they are now exposed. So the sperm becomes active and it swims faster. It will be able to penetrate the secondary oocyte. So this process of removal of the deposits on the surface of the acrosome is called capacitation. And this happens after 5 to 6 hours after insemination. After the semen is deposited, after 5 to 6 hours the capacitation starts. Apart from this, the secretions of the male reproductive accessory glands, as we have seen, seminal vesicle and bulbourethral glands, these secretions, they protect the spermatozoa from the acidic condition which is prevailing inside the female genital tract. And now let us see about the physical and chemical effects of fertilization. After fertilization, there are some physical as well as chemical changes happening. What are they? The first is the acrosomal reaction. This is caused by the capacitated sperm. The capacitated sperm, that is after capacitation, the sperm, it releases some chemicals, a group of chemicals. These chemicals are collectively called as sperm lysine, and it includes hyaluronidase, corona penetrating enzyme, zona lysine or acrosome. Hyaluronidase, what it does is, it digests the ground substance which is binding the follicles. You see, this is the oocyte, it is surrounded by the follicles, granulosa cells. These cells are held in place with the help of cementing substance. Okay. This hyaluronidase, it digests the cementing substance so that the follicles become free, so the spermatozoa can enter into it. The next one, corona penetrating enzyme. As the name indicates, it penetrates the corona radiata which is present. And the third is zona lysine, also called as acrosome. It digests the zona pellucida. And for this process to happen, calcium ions are important. So if there is deficiency of calcium ions, the penetration of the sperm into the secondary oocyte may not happen. Next, in the acrosomal reaction, the plasma membrane of the sperm and the secondary oocyte, they fuse together, and that results in a depolarization of the secondary oocyte. This depolarization prevents the entry of other spermatozoa. Thereby, it stops or prevents polyspermy. So, the depolarization ensures prevents polyspermy, because why this polyspermy is not required? See, the spermatozoa it has haploid number of chromosomes and the egg has haploid number of chromosomes. When they fuse together, they form a zygote which is diploid. Just imagine another sperm is also entering. That will result in a triploid condition and the embryo may not survive. So nature doesn't want this to happen. It wants to protect that embryo. So that's the reason polyspermy is prevented. The next stage is that is next to acrosomal reaction, we have the cortical reaction. See, if you look at the oocyte, there are cortical granules which are present just below the plasma membrane. Cortical granules are present. These granules, they fuse with the plasma membrane. See, this is a granule. I'm making it a little bigger. So this goes and it binds with the plasma membrane and it will empty its content here. Okay. So this is in between the plasma membrane and the zona pellucida. This is zona pellucida. This is the plasma membrane. So the granules are releasing their contents in between these two phases. And after some time, this part it gets hardened. It gets hardened. And thereby it prevents entry of another sperm. See, all these acrosomal reaction, cortical reaction are happening after the entry of one single sperm. So now what is required is done. So further entry of sperm is prevented. So let us see what happens in the sperm entry. At the point of contact of the sperm, the secondary oocyte, it produces a cone-like structure. And it receives the sperm. So the secondary oocyte is aiding the entry of the spermatozoa to enter. So this fertilization cone, it helps in receiving the sperm. At this point, you remember that the spermatozoa, it has two centrioles. One is proximal, one is distal, and another one, the third one is the ring centriole. This proximal centriole, it gets divided into two. So that when the sperm enters into the oocyte, secondary oocyte, later for cell division, these secondary centrioles will produce the spindles. The oocyte, secondary oocyte does not have a centriole. That is contributed by the spermatozoa. And next is the karyogamy. As the name indicates, karyo means nucleus, gamete means fusion. So this stage involves the fusion of the male nucleus and the female nucleus. So once the spermatozoa enters the secondary oocyte, the secondary oocyte completes the second meiotic division. So far it was in the prophase 1 stage. So now after the entry of the spermatozoa, it has completed the second meiotic division and now it becomes an ovum. And as usual, one polar body is also formed. Now what happens? The sperm, as you know, it has head, middle piece and tail. Neck is also there. So the head, it gets separated from the rest of the body. So the head alone, it gets separated. So that contains the nucleus. That nucleus, now it is called as the male pronucleus. And already there is another nucleus, that is the nucleus of the oocyte, which is called as the, now it is called as the female pronucleus. So now both male pronucleus and female pronucleus are there. And what happened to the polar body? What happened to the tail? They get disintegrated. And Now the male pronucleus and the female pronucleus, they join together. They form a zygote, which is diploid. Next is the activation of the egg. As the sperm enters into the egg, it stimulates the metabolism of the zygote. So earlier it was a secondary oocyte. Now it has become a zygote. The entry of the sperm stimulates the metabolism like cellular respiration, protein synthesis and all. So it starts growing. And there is a theory which was proposed by F.R. Cleary, fertilizing and anti-fertilizing reaction. He proposed that the egg produces a substance on its surface which is called as fertilizing, and the sperm produces another substance on its surface which is called as anti-fertilizing. It is the interaction between these two, fertilizing and anti-fertilizing, which brings the fertilization, that is the fusion of the male and female gametes. We have seen that during fertilization, the pro-male nucleus and the pro-female nucleus, they fuse together and they form a zygote. This is also called as aphemesis. After that, what happens? After that, the zygote undergoes cleavage. Cleavage means it is a sequence of mitotic divisions. There will be repeated mitotic divisions which will convert the single cell zygote into a multicellular blastocyst. That is called as cleavage. It is a series of mitotic divisions which will convert a single cell zygote into a multicellular blastocyst. Now let us see the process involved in this. See, in the fallopian tube, in the ampulla, the zygote is formed. After the formation of zygote, the first division, it happens after 30 hours and it results in two cells and they are called as the blastomeres. Second division happens in the 40th hour, that results in 4 cells. And the third division, it takes place in the third day and it results in a solid mass which is called as a morula. I will be showing you some diagrams which will give you a clarity about what's happening during this process. And this cleavage in human being, it is called as holoblastic cleavage. Holoblastic means if this is the zygote, it involves the entire egg. This is holoblastic. Because in some zygotes, you can see there will be a lot of food yolk present in the egg. So the division will not be happening fully. So that is not holoblastic. But here it is holoblastic because it involves the entire egg. Now let us see the significance of cleavage. Significance of this cleavage. See, it helps in the distribution of the zygote's cytoplasm among the blastomeres. Secondly, the mobility of the protoplasm of the zygote during cleavage, it facilitates morphogenetic movements within the zygote. Morphogenetic movements within the zygote, which results in the formation of the germ layers. And the third is, it restores the nucleocytoplasmic ratio. What is nucleocytoplasmic ratio? If this is a cell, it has a nucleus and this is the cytoplasm. So this nucleus can control the entire cell. But when the cell grows, suppose if the cell is becoming like this, that is, it has grown and the nucleus is this. Now the amount of cytoplasm is too much, so this nucleus, it cannot manage. So the cell won't survive. So in nature what is happening is, when the cell grows, it is divided into two, and each one is provided with one nucleus. So the nucleus and cytoplasmic quantity ratio is maintained. So this is done by cleavage. So in this diagram you can see where the fertilization occurs and how the cleavage is happening, where it is happening, and what happens to the morula. Those things are clearly depicted here. So this is the ovary. This is the ovary, and here you can see the corpus luteum, and the mature follicle is released. And once it is released, here it is released, it is captured by the fimbriae, which is present in the infundibulum of the fallopian tube. And then it is driven inside the fallopian tube with the help of the ciliated cells which are present. So here you can see the corona radiata of the ovum, the zona pellucida, nucleus and cytoplasm. So this is the ovum. And in day one, the sperm approaches, many sperm approaches, and only one is able to penetrate in. And after shedding the middle piece and the tail, it becomes the sperm becomes the male pronucleus, and here is the female pronucleus. In day two, they join together to form the zygote, and here you can see the first cleavage happening. The first cleavage happens and it results in two blastomeres. And meanwhile, you can see the egg is moving towards the uterine cavity. And see here, second cleavage, 4 cells are present, and then the third stage, morula. Here, a solid mass is formed, which is called as the morula. So by this time, almost it has reached the oviduct where it is joining the uterus. Then this morula is converted into blastocyst, and later this blastocyst it gets attached with the endometrial wall. So the implantation happens here. This diagram, it shows the early stages of embryonic development, how the zygote is converted into a blastocyst. So this is the zygote. Here you can see the zona pellucida, which is the outer layer, and here you can see the nucleus, and here are the polar bodies. And in the 2-cell stage, that is after the first cleavage, it results in 2 cells, and these cells are called as the blastomeres. And here you can see the zona pellucida and the polar body. Next is the 4-cell stage, which means that the second cleavage is completed and it results in 4-cell zygote. Then this 4-cell, it undergoes the third division and it becomes a 8-cell stage. Here also you can see the zona pellucida, the blastomeres, 8 blastomeres are inside. And next is the 16-cell stage. Here it gets the appearance of a solid structure, so it is called as a morula. And then it is gradually converted into a blastocyst. So in the blastocyst, you can see the zona pellucida is breaking. So it is getting broken, and here you can see an outer layer of cell which is called as a trophoblast. And there is an inner cell mass attached to one part, and this is the inner cell mass. And the cavity formed is called as a blastocyst. So the morula is converted into a blastocyst. In the blastocyst, it contains 2 structures. One is a outer membrane structure. This is called as a trophoblast. And within the trophoblast, there is a mass of cells which is attached with the inner side of the trophoblast. And this is called as the inner mass. The region to which the inner mass is attached, this side it is called as the animal pole. It is called as the animal pole. The inner mass, it develops into the future embryo. Inner inner mass, it proliferates, it undergoes changes, morphogenetic changes, and it develops into an embryo. Whereas the trophoblast, it forms the extra embryonic membranes like corium, allantoin, all these membranes are formed by the trophoblast. And the trophoblast, where it is getting attached with the inner mass, this part, the area of the trophoblast which is getting attached with the inner mass of the cell is called as router. So once the blastocyst is formed, next what happens is the attachment of the blastocyst with the endometrial wall. So that stage is called as implantation. So implantation is the attachment of the blastocyst with the endometrial wall of the uterus. And this happens after 7 days. 7 days after fertilization, implantation happens. And here, the region of contact, this area, okay, the region of contact of the trophoblast with the uterus, it gets differentiated into 2 layers. So the trophoblast, at the point of attachment with the uterus, if this is the uterus, the trophoblast, it goes and attaches. In this region of contact, it gets differentiated into 2 layers. One is a syncytial trophoblast, and the other layer is a cytotrophoblast. Syncytium means many nucleus. So this area, it is not made up of cells. Okay, there is no division of cells in this area, but it contains a lot of nucleus. Whereas this cytotrophoblast, it is made up of individual cells. And what happens if this is the endometrium, the blastocyst, it sinks into the endometrium. So if this is the endometrium, the blastocyst, it gets sink into the endometrium. And after sinking, it forms bill-like, finger-like structures which penetrate into the uterine wall. Within the inner mass, the cells are differentiated into 2 layers. One is the hypoblast and the other is the epiblast. So this diagram you can see how the blastocyst gets implanted in the endometrial wall. So this is day 6. This is the blastocyst, and you can see the trophoblast, the inner mass. It comes and binds here. Here, we should know the role of zona pellucida. The zona pellucida, it covers the blastocyst and it prevents the attachment of the blastocyst in an improper site. Only when the blastocyst reaches the proper site, then the zona pellucida, it exposes the receptors which are present on the surface of the trophoblast, so that this can attach with the blastocyst can attach with the endometrial wall. So here you can see the structures of the endometrium of uterus. Here is the connective tissues, these are the connective tissues, and this is the uterine epithelium. And as I said earlier, on the 7th day, the blastocyst, it gets implanted. So first what happens is the blastocyst, it starts sinking into the endometrium. Here you can see it is getting sinking, and at the area of contact between the trophoblast and the uterine wall, the trophoblast, it gets differentiated into 2 layers. One is the syncytial trophoblast. Here you can see there are many nucleus, but it is not divided into cells. And this is the cytotrophoblast. These are cellular. So this is cytotrophoblast, which is cellular, and this layer which looks like amoeba in the diagram, it is a syncytotrophoblast. And apart from this, we have also seen that the inner mass, it starts getting differentiated into 2 layers. one hypoblast and one epiblast. We will see a detailed structure of this now. So here you can see how the embryonic disc is formed. So this is the blastocyst with the inner cell mass. This is the trophoblast. Later what happens, this inner cell mass, it gets differentiated into two layers. One columnar epiblast. Epi means above. So one columnar epiblast layer and a hypoblast layer. Soon a cavity will appear. Here you can see this cavity. It is called as the amniotic cavity. It is formed above the epiblast, and on the roof of this cavity is the amniogenic cells, and the floor of this cavity is made up of epiblast. And later a yolk sac is also formed. Yolk sac, here you can see the yolk sac. So in this diagram you can see that the primary yolk cell, it has increased in its size, and the trophoblast, it forms the extra embryonic mesoderm. Here, extra embryonic mesoderm. And later what happens, this mesoderm, extra embryonic mesoderm, it gets differentiated into a inner splanchnopleuric extraembryonic mesoderm, this part, which is immediately next to the yolk sac, and outer somatopleuric extraembryonic membrane, this part, and between these two is the extraembryonic coelom. And this diagram you can see the yolk sac is now called as a secondary yolk sac, because it is compressed by the growth of these splanchno and somatic pleuric extra embryonic mesoderm, and by the coelom it's forming, extra embryonic coelom. Because of this, it gets reduced in size. So here you can see the extra embryonic membranes. This is the amnion, which is circled here, and this is the chorion, and this is the secondary yolk sac. Right. Next we are going to start a study about the extraembryonic membranes. So before doing that, let us have a look at the diagram so that you will have an idea. So in this diagram you can see the center is the embryo, this black color area. This is the developing embryo, and this is the anterior end of the embryo, and this is the posterior end of the end of the embryo. And this white color, it is the digestive tract, the developing digestive tract. And from this digestive tract you can see a sac-like structure extending outside, and this is called as the yolk sac, and it contains the splanchnopleuric extraembryonic mesoderm, and it has an endoderm. So these two structures, it forms the yolk sac. And you can also see at the posterior end there is another sac-like structure which is extending outside, and this is called as the allantois. Allantois is also similar to yolk sac. It is also formed by the same membranes, splanchnopleuric extraembryonic membrane and the endoderm. So now we have seen what is yolk sac and what is allantois. Next, let us see what is chorion. Chorion, here it is made up of the trophoblast, which is the outermost layer, what we have seen in the blastocyst, and it is also having the somatopleuric extraembryonic membrane. So unlike yolk sac and allantois, here it is made up of the trophoblast and the somatopleuric extraembryonic membrane. So let us have a look at where it extends. So we can start from here, that is this entire area, and this entire area. This together it forms a chorion. So it almost covers the entire embryo, the amniotic membrane. Only a part of the yolk sac is outside. It's lying outside. And the next part is the amnion. So the amnion is made up of, again, somatopleuric extraembryonic mesoderm and the trophoblast. So this grey color area, it is called as a amnion. And this amnion is filled with the amniotic fluid. And here you can see that the chorion, it gets extended into finger-like structures. Okay, the villi, and the placenta will develop in this region. So as I said earlier, now let us see about the various extra-embryonic membranes. See, these extra-embryonic membranes, they protect the embryo. They protect the embryo from desiccation because it provides a fluid surrounding. It prevents the embryo from getting dry. And this fluid, it gives buoyancy to the embryo. So it protects the embryo from mechanical shock, and it also helps in the absorption of nutrients and gases. So first we'll see about the amnion. So this is the amnion and this is the amniotic cavity. It is a double layered structure. It is a translucent membrane. It is filled with the amniotic fluid. This amnion and its fluid, it provides buoyancy and protects the embryo from any injury. Apart from this amnion also helps in regulating the temperature of the fetus, and it acts as a medium for the fetus to move around. The fetus can move within the amniotic cavity in the fluid. Next is the yolk sac. The yolk sac, it forms a part of the gut. As we have already seen, it arises from the gut. It forms a part of the gut, and it is the source of earliest blood cells and blood vessels. Next is the allantois. Here, allantois. It forms a small out-pocketing of the embryonic tissue at the caudal end of the yolk sac. And it is a structural basis for umbilical cord. The umbilical cord actually connects the embryo to the placenta. This allantois, later it becomes a part of the urinary bladder. Then we will see about the chorion. The chorion is the outermost membrane, and it encloses embryos and all other membranes, and it helps in the formation of the placenta. The trophoblast, it secretes the HCG hormone. Now let us see what it does. So HCG is produced by the trophoblast. Its function is very similar to the luteinizing hormones. That is, it maintains the corpus luteum. And it stimulates the corpus luteum to secrete progesterone. You know that the endometrium is maintained by the progesterone. So, for the successful survival of the embryo, the endometrium needs to be maintained. So that is done by progesterone. So because progesterone is produced and the endometrium is maintained, it prevents the formation of menstruation. There won't be any menstruation during the pregnancy. Apart from this HCG, it makes the cervix of the uterine to produce more mucus. So what happens, see, cervix means neck. If this is the uterus and here is the cervix, the extra mucus, it gets accumulated and it forms a plug-like structure which protects the embryo. And now let us see the difference between what is embryo and what is fetus, because many of us have a confusion. See, embryo means it is the early stage of development. In human, from the period of conception, from the time of conception till the 8th week, we call it as embryo. And fetus means after the 8th week, till the birth of the young one, it is called as fetus. So fetus means it is an unborn young one. And gastrulation, just to define what is gastrulation, it is a process by which blastocyst is converted into gastrula by the rearrangement of the cells. The blastula, within the blastula, there is rearrangement of cells and it is getting converted into gastrula. Here the cells move, and this movement helps in developing different morphological structures in the developing embryo. Because of this, these movements are called as morphogenic movements. That is, morpho means structure, genic means giving rise to. Since these movements give rise to different structures, these are called as morphogenic movements. And in human, there are 3 germ layers: ectoderm, endoderm, and mesoderm. So it is called as triploplastic. Human embryo is triploplastic and it has 3 layers. Let us see what these 3 layers develop. So these 3 layers are called as the primary germ layers, and they act as primary tissues. The first layer, ectoderm. Ecto means outside, derm means skin. So this develops into the nervous system, the central nervous system, peripheral nervous system. Apart from this, it also gives rise to the mammary glands. And since it is ectoderm, it gives rise to the epidermis. Next, mesoderm, which is the middle part of the 3 layers, that develops into the urinogenital system, and all connective tissues they arise from the mesoderm. It also gives rise to the cartilages and bones and muscles. The endoderm, endo means inside, derm means skin or layer. So the endoderm, it gives rise to all the internal organs like liver, pancreas and parathyroid. And it also gives rise to the epithelium of GI tract as well as the respiratory tract. So now we have seen what are the development of the 3 primary germ layers. Now let us see about the human pregnancy. The human pregnancy, it lasts for 280 days or 40 weeks. This period is called as gestation period. And it is divided into 3 trimesters. For better understanding, it is divided into 3 trimesters. And in the first trimester, it is a major period for organogenesis. Most important organs like in the heart, lungs, liver, limbs, and the external genitalia, it arises in the first trimester. And in the second trimester, the face is well formed, the eyelids, eyelashes have appeared, and the baby will start blinking the eyelids. Apart from this, the body is covered with fine hair. Throughout the body is covered with fine hair. The muscle tissues develop well. The bones, they get harder. And in the third trimester, the baby is fully formed and it is ready for delivery. Placentation is the formation of placenta. What is placenta? Placenta is an intimate connection between the fetus and the uterus. So it is an intimate connection between the fetus and the uterine wall. The chorion, on its outer surface, it produces finger-like extensions. This is the chorion. it produces finger-like extensions on the outer side. And these are called as the villi. These finger-like structures are called as the villi. These villi, they grow into the tissues of the uterus. They penetrate the uterus tissue and they form the placenta. And in human it is called as chorionic placenta because the placenta is formed by a roll of the chorion. So it is called as a chorionic placenta. And the placenta, it has two counterparts. One is the fetal part, the other is the maternal part. The fetal part is the chorion, and the maternal part, that is the part given by the mother, is the decidua basialis. It is a membrane-like structure. And the villi which has penetrated into the uterus, they are bathed in the flood of the mother. They are bathed. This contains the blood of the mother. These villi are bathed with it. How this is happening? It is happening by the erosion of the uterine mucosa. The mucosa of the uterus is eroded, as well as the epithelium is eroded, connective tissues, endothelium, all are eroded. So there is blood accumulation in which the chorionic villi are bathed. In human, the placenta is also said to be hemochorial placenta because of the intimate connection between the fetus and the mother. Both they take part in the formation of the placenta and they are intimately connected. So it is called as hemochorial placenta. And the placenta, it is connected with the umbilical cord. And the umbilical cord is interconnected with the mother tissue. So the umbilical cord acts as a connecting bridge between the placenta and the mother cells. And this placenta of human being, it is called as metadiscoidal placenta because the villi which is penetrating into the mother tissue, it is restricted to a spherical area, restricted to a disc-shaped spherical area. So it is called as metadiscoidal placenta. So in this diagram you can see this is the umbilical cord, and the umbilical cord runs over, it bifurcates, runs over the placenta. This is the placenta. It's a spherical disc-like structure. Placenta plays a vital role in developing the embryo. Let us see the functions of placenta. See, the embryo has to grow. For that, it has to be fed with nutrients. So that nutrition provided is being done by placenta. Apart from that, it also provides the fetus with gases like oxygen. So it helps in respiration. And moreover, there will be some waste formed within the fetus environment. These wastes are removed by the placenta, so it helps in excretion. Apart from that, it acts as a barrier. Everything from the mother, it cannot enter into the fetus. So it acts as a barrier, especially it prevents the entry of teratogens into the fetus. What are teratogens? Teratogens are substances which will cause abnormal morphology in the fetus. So such substances cannot enter into the fetus because the placenta, it acts as a barrier. The placenta, it also acts as a temporary endocrine organ. It produces human chorionic gonadotrophin, human chorionic somatotrophin. It is also called as human placental lactogen and relaxin. All these are formed by placenta. So regarding the functions of HCG, we have already seen it acts more or less similar to progesterone. It activates the corpus luteum. It induces the corpus luteum to produce progesterone. Thereby, the maintenance of endometrium is done and menses is prevented. Coming to human chorionic somatotrophin, it induces the development of the mammary glands. And relaxing, it plays a vital role in parturition. It facilitates parturition. It softens the connective tissues in the pubic symphysis so that the baby can pass easily through the birth canal. Apart from this, there are many other hormones whose level are increased during pregnancy, like estrogen, progesterone, cortisol, prolactin, thyroxine. They are normally there, but in the case of pregnancy, they are produced in large quantities just to support the development of the fetus. This diagram, it shows a clear picture about how the placenta, umbilical cord and the maternal tissues are interconnected. So this is the placenta, which is a disc-shaped structure. And from this arises the umbilical cord. You see, the maternal portion of the placenta, it is the decidua basalis, this part, on this part. And this is the fetal part. This is formed by the choria. First let us see the maternal part. So here, the maternal endometrial arteriole, it passes through these tissues. And here it empties its blood in this area. And then this shows the intervillous space containing the maternal blood. So the villi are bathed in the blood of the mother. And this shows the maternal endometrial venule, which takes us out the waste materials. So that's with the maternal part. Coming to the fetal part, that is a chorion. Here you can see the finger-like projections which are penetrating inside, which are called as the chorionic villi. And this is the fetal blood vessel. You can see the arrows indicating the direction of the flow of blood. So from the maternal blood, the nutrients and other things are taken by the fetal blood vessels and it is carried through the umbilical cord, through the umbilical arteries. And this is the amnia. This is the mucous connective tissues. The umbilical cord contains 2 umbilical arteries and 1 umbilical vein. The next stage is parturition. Parturition, it means the expelling of the baby, and certain structures related to the embryonic membrane, they are called as afterbirth. So the expelling of the baby as well as the afterbirth, it is called as parturition. And this process is induced by the nervous system as well as the endocrine glands of the mother. Throughout the gestation period, the mother will be experiencing periodic contractions, mild contractions. And they are called as Braxton Hicks contractions. These are not labor pain, often they will be confused with the labor pain. These are false labor pains. They will be happening periodically at a mild level. What happens during parturition is the fetus initiates the first step. The fetus and the placenta, they stimulate the oxytocin secretion by fetal ejection reflex. Fetal ejection reflex. It stimulates the release of oxytocin. This oxytocin, before labor pain, it is produced in large amount. So that leads to the contraction, vigorous contraction of the uterus. Apart from oxytocin, as we have seen already, relaxin also plays a major role. It brings about the flexibility of the pubis symphysis. It relaxes the ligaments of the sacroiliac and sacrococcygeal joints, which will ease the pain. And This dilates the cervix of the uterus. So the uterus neck now it has got dilated. There is one hormone which has been recently found, that corticotrophin releasing hormone. It is there, but its role in parturition has been recently established. This is produced even in normal women, but in normal women it is produced in the hypothalamus. Whereas in pregnant women during parturition, this is produced by the placenta, and it is believed to be the clock which determines when the delivery is going to happen. For example, women with lesser amount of this CRH, they experience a late delivery, that is delivery after the due date. And women who are having a higher concentration of CRH, they go for premature delivery, that is the baby is born before the due date. Labor pain is a pain which is experienced at the time of delivering a baby. And this labor, it has 3 stages. The first stage is the stage of dilation. So with the onset of the labor, the dilation of the cervix will occur. That is, the neck of the uterus will dilate. And for this dilating process, it may take some 6 to 12 hours. It varies with individuals. And there is regular contraction of the uterus. The uterus will regularly contract. And now, because of the contraction, the amniotic sac, it ruptures. Because of that, the amniotic fluid will start flowing through the vagina. This is an indication that delivery is going to happen. The second stage is the stage of expulsion. In this stage, the baby is expelled out of the body. It may take 10 minutes or it may take several hours. That also varies with individuals. And the baby passes through the neck of the uterus, that is the cervix, and then through the vagina, and then it is delivered. The third stage is the placental stage. It is the time between, that is, once the baby is delivered, then after some time, because of vigorous contractions of the uterus, the afterbirth means the placenta and other things, other extra embryonic membranes, they will be expelled out. And that's happening because of powerful contraction of the uterus. And these contractions, it is not only the uterus is contracting, it also constricts the blood vessels so that the extra embryonic membranes could be easily expelled out and hemorrhage could be prevented. And after delivery, within some 28 to 35 days, the uterus will return to its normal size. Lactation is a process by which milk is produced. At puberty, the hormones estrogen and progesterone influence the development of mammary glands. But the secretion and storage of milk will start only after delivery. Maybe within 24 hours, it is influenced by the hormone prolactin. So because of the action of prolactin, the mammary glands will start producing milk after delivery. The ejection of the milk is controlled by oxytocin, and this process is called as let-down reflex. So oxytocin, it helps in ejection of the milk. Oxytocin also plays a role in bringing back the uterus to the normal size. And a pregnant woman may produce in a day 1 to 2 liters of milk. Colostrum is a technical term given to the milk produced in the first 2 to 3 days after delivery. So colostrum is a yellow colored fluid which is rich in lactoalbumin and lactoproteins. The colostrum also contains a type of immunoglobulin, that is IgA. This IgA it passes from the mother through mother's milk to the fetus, to the baby. And within the baby, it provides immunity to the growing baby. So it produces passive immunity in the baby. Coming to the composition of milk, it contains large amount of water apart from the inorganic as well as organic substances. The main constituents are fat. Fat exists as droplets, fat droplets, and the protein, casein and sugar, lactose. These are the main constituents. Apart from this, it contains mineral salts like sodium, calcium, potassium, phosphorus and small amount of vitamin C. But milk is very poor in iron content. The regulation of milk secretion is mainly regulated by the nervous system. Apart from that, the psychic condition of the mother and the hormonal level of the mother. Breastfeeding is very important for the health of the baby as well as for the mother. Let us see the importance of breastfeeding. See, the milk, it contains an inhibitor peptide. So if the mother is not going to feed the baby, this inhibitor peptide will accumulate within the mammary glands and it prevents further secretion of the milk. So it is a negative feedback inhibition. So if the mother is giving milk, this peptide will get diluted and it will go off and again milk will be produced. But if she is not going to give for a period of time, this peptide, inhibitor peptide, will inhibit the formation of milk. And feeding, mother feeding, it is a method of contraception. Because as long as the mother is feeding the baby, the menstrual cycle won't happen. So it is also a method of birth control. And the World Breastfeeding Week, that is August first week of August. This is promoted by the World Alliance for Breastfeeding Action, WHO and UNICEF. These together, they are promoting this to stress the importance of breastfeeding during the first 6 months regularly and supplemented breastfeeding for the next 2 years. So to promote this only, this World Breastfeeding Week is organized. It gives good health to the baby as well as mother feeding. It guards the baby from neonatal jaundice, pneumonia and cholera. Tamil Nadu government has also taken various efforts like various projects like Mother's Milk Bank, feeding rooms in bus terminal and organizing camps on the first week of August to promote breastfeeding. This name, Arunachalam Muruganandan, it is a name which stands representing India on women's menstrual hygiene. He is the first person to design a first low-cost machine. He was motivated or in fact driven by the fact that 70% of reproductive diseases in India, it's happening because of poor menstrual hygiene. And 23% of girls who attend puberty, after puberty, they become dropouts of schools, 23%. So, he started his research in 1999. He worked for 5 years and in 2006 he made his presentation about his machine. He presented the low-cost machine for making sanitary napkins in IIT Madras on a national competition for innovation. And out of the 943 contestants, he stood number 1. And within 18 months period, he has made 250 machines which have been supplied to Bihar, UP, MP and Rajasthan. And in 2014, the Times magazine has selected him as one of the top 100 Indians who are the most influential people. And in 2016, he was awarded Padma Shri. Now let's see few terminologies related to this topic. First is cesarean. Cesarean means it is making a surgical incision in the abdomen and in the uterus of the mother to take the baby out. This will be done in conditions when the baby position is incorrect or maybe because of the health conditions of the mother. And cryptorchidism. Crypto means hidden and tropism refers to the testis. See, in human being, in a male child, the testis has to descend into the scrotum. As we have seen already, the scrotum, it needs an optimal temperature, not very high, not very low. So it is, the testis is kept in the scrotum so that the scrotum can move up and down and regulate the temperature of the testis. But in few male babies, maybe 1 to 3% of the male babies born, the testis won't descend into the scrotum. So that condition is called as cryptorchidism, and that has to be corrected surgically or else they will not be able to produce viable sperms. Sterile, the word sterile means the inability to produce viable sperm. And ageospermia, it refers to the condition where the semen will not have any spermatozoa. And archidectomy, it refers to the surgical removal of testis or castration. It is a kind of male contraception. And spermac, it refers to the first ejaculation of the semen. And menarche refers to the first menses in women.